Hello everyone, today we're going to go over the lecture Introduction to Eukaryotes and Prokaryotes. And once I get started with sharing my screen, I'm going to minimize my camera and turn it down so it's facing the table so it doesn't make that irritating clicking sound. All right, and I have a background behind me today that is from a small painting that was done by my cousin for me. I like to think about being down by the ocean when I'm tired or stressed out. So it's beautiful, beautiful background for me. Getting the slideshow started. We're going to start by talking about cells in general, and then make distinctions between prokaryotic and eukaryotic cells. And then we're going to look at just bacteria, not archaea today, and have an introduction to some of the taxonomic groupings within the eukaryotes. Not the major ones, we're going to come back to those in lots of later labs, but we're going to look at some of the diversity of eukaryotes. So when we talk about a cell in general, a cell has several characteristics that they share. They have to have a cell membrane, DNA is a genetic molecule. They have to have ribosomes in order to interpret the genetic information, and they have cytoplasm, liquidy stuff inside. So these are our shared characteristics of all cells. Cells, and indeed all things that we define as life, also have these characteristics that we call charm, one way of summarizing them. They have complexity or organization. They have homeostasis. They have adaptation. And they do reproduction. And they do metabolism. So these are the shared characteristics of life in terms of the functions. Homeostasis and metabolism, for example, basically staying alive and using energy and molecules by maintaining constant internal environment. And then the structures that define a cell. So multi-celled organisms are made up of many, many cells, but each cell has to have these four characteristics. We start looking at the prokaryotic kind of cell versus the eukaryotic kind of cell. We see that there's two major types of differences between them: size and complexity or structure. So the size for prokaryotes is going to be small. They're going to be 1 to 10 microns. So microns are micrometers, 1,000th of a meter. Eukaryotic cells are large, and that's 10 to 100 microns. The structure of prokaryotic cells is simple, whereas for eukaryotes it's complex. And the major way that this simplicity versus complexity manifests itself is that prokaryotic cells have no membrane-bound organelles. And eukaryotic cells do have organelles. So a membrane-bound organelle just means that it's an organelle or a substructure that's partitioned inside the cell. So basically, this difference is like saying that prokaryotes don't have organelles, whereas eukaryotes do. So organelles are things like the nucleus, things like the mitochondria and chloroplasts, things like the Golgi apparatus, endoplasmic reticulum, and vesicles and vacuoles. So it's like the difference between a big house with lots of rooms in it that are divided by panels, or a studio apartment that's just one space that's open on the inside. So on the inside of prokaryotes, you do find DNA, but you don't find it neatly stored inside of a nucleus, especially for that purpose. Eukaryotes have DNA, but you find it inside the nucleus. All right. Okay. We're going to look at bacteria in this lab today, and we are only just very, very lightly going to scratch the surface of the interesting and complex ecology of bacteria. So on this diagram, they're called eubacteria, the true bacteria, whereas the archaea are called archaebacteria. And we can see that the rest of the taxa on the slide are the eukaryotes. So eukaryotes have relatively recently been regrouped into these different clades. And today we're going to look at the SAR clade, and capital S, capital A, capital R is not a word that has a meaning. It is to indicate that this clade is formed of lumping together the stramenopiles, the alveolates, and this taxon, which is called Rhizaria. So that's the R. So we have the SAR clade, and then we have the archaeplastids, and archaeplastids are going to include in this taxon the land plants, right? So there's a tree here that's showing us that our large multi-celled land plants are in this. So we'll come back to the archaeplastids. We'll spend a few labs on them and mostly on land plants. Today we're also going to talk about some examples of the excavates, excavata. And we're going to come back to these two groups over the rest of the semester because taken together, the amoebozoa and the opisthocons are in the taxon called uniconts. Not unicorns, uniconts. So we see that within the opisthocons, we have fungi, one of our other former major kingdoms. We have the metazoa here, which is animals. And then we can see on this cladogram that we have the taxon that is a sister taxon to animals is this Choanomonada, but it's also called choanoflagellates, and it's a single-celled organism. And also the fungi, these nuclearia, are a single-celled taxon. So the sister taxa to these groups where we do have multi-celled organisms are single-celled. So that means that multicellularity, or being multi-celled, evolved on each of these different branches separately. So if we think about being multi-celled as a characteristic, and we are talking about land plants, fungi, and animals, is being multi-celled homologous or analogous? Right, it evolved separately in these different groups. So that means it's analogous because they don't have a recent shared ancestor that's also multi-celled. All right, so we'll come back to those other taxa later in the semester. We're going to do bacteria, SAR, and excavates today to look very, very quick glimpse into the diversity of those groups. All right, so this is our first observation for lab today, and it's intended to give you a good relative size comparison between eukaryotic cells and prokaryotic cells. We'll talk about how big a difference there is in size, but let's really bring it home by seeing a picture of it. So these cheek cells have been stained with a blue stain. They've scraped from the inside of some poor person's mouth. And we can see that they're eukaryotic cells because they have a very obvious and dark staining nucleus that we can see clearly. So the prokaryotes on this slide are also possible to see because of the staining. We have an example here that's a bacteria. So whether you brush your teeth really well or not, you're still going to have bacteria in your mouth. So we always get some bacteria on these slides as well. So we can see that the bacteria is, 1,000 times smaller than the eukaryotic cell. So that makes it really easy to see. When we're talking about the archaeobacteria and eukarya, we want to make some comparisons between them, looking at what kind of cell they have, whether they can be single or only multi-celled, whether they have a cell wall, and whether they can do locomotion or not. So this is just a quick summary slide that I stuck in here extra. You can write this down in your notes. And both archaea and bacteria are going to have a prokaryotic type of cell, whereas eukarya are going to be eukaryotic. We have a hard time remembering which is which. The prokaryotic cells are simpler, so you can think of they're like a prototype for the more complex cells that came later. Archaea, any kind of prokaryotic cell, so archaea and bacteria, can only be single-celled. And eukaryotic organisms can be single or multi-celled. Archaea and bacteria both have a cell wall. It's made of different molecules, but they're a yes. Come on, pen. Yes, yes, they always have a cell wall. Eukaryotic groups differ in whether they have a cell wall. So there are yeses or no's. So each taxonomic group is either going to have a cell wall or not have a cell wall, but we have many different examples of groups within the eukaryotes that have one or the other. For locomotion, archaea are a no. Bacteria are a yes, but the only structure that they have for that is a flagellum. Eukarya, when we have single-celled organisms, they have some options here. They can have cilia, they can have flagella, or they can use some kind of pods. All right, so we say they have pseudopods, and that's some extension, sort of a lobed or globby extension of the cell like amoeba has that allows them to crawl. Right, so cilia and flagella, they're going to be able to swim with those. Pseudopods, they're going to be able to crawl. If we have a multi-celled eukaryotic organism, some don't have locomotion, like a tree, whereas animals are going to have locomotion with muscle tissue and a skeletal structure. So we'll come back to those later on in the semester, really talking about single-celled eukaryotes here. So remember, I often talk about putting things into categories and knowing the characteristics of them based on those categories. This is a great example of the kind of thing I'm talking about. You should know these groups and the characteristics that they have. So when we start talking about the bacteria, we're just going to look very, very small amount of detail at them today. There's only a few things I'm going to expect you to know. And these are things that we can see under light microscopy, that is with a compound microscope, not with an electron microscope. So bacterial cells, they have a cell wall, and they're going to come in 3 different shapes. They're going to have either a round shape, and these are pretty tiny. They can have this oblong or rod shaped. These are sort of medium sized, and they have this oval shape, and they're kind of thick. Right, the spirili are long and thin. You can't always see that they're spirally, like a spiral, like a rotini pasta, but they're going to be a lot longer and thinner than a bacillus. Now, cocci and bacilli can be tricky because, as we can see on this figure that's lower down on the page, that we have some rod-shaped bacteria here, but they're chained together into a colony. And so we might think it's a long skinny cell, but in reality, we have to look carefully and try and see those little separations between the cells. We also want to scan around the slide and look for ones where there's an individual that's separate. If we see one where there's an individual that's separate, it's clearly not a broken piece of something, that it's going to help us know what shape those bacteria are. So we'll see some that are clumped together. We want to ignore those. Look for ones that are separate. Right? So you want to scan around on your slides or look around the pictures to find something. Now, the other characteristic that we can pretty easily determine using light microscopy is whether they have a certain characteristic of their cell wall that determines how they take different stains. Right? So if you think about it, if you're doing some tie-dyeing at home, you want to get a cotton t-shirt in order to do that, because that cotton fiber is going to take the stain much better. You're the dye, you aren't going to try and use a polyester shirt, because it just isn't going to stick. The dye can't bind to it. So same thing happens with different types of bacteria. It turns out there's some taxonomic importance to whether they respond to a particular stain. And the doctor that invented it is called Graham. And so this technique is called Graham staining. And it's a two-step process. The first step uses a red dye. And then the second step is to add a purple dye. And then the specimen is washed. So every bacteria is going to take up that red dye or pink dye, and then only the ones that are gram positive, that is, they have a particular structure, particular molecules in their cell wall, are going to absorb, and the violet stain is going to stick to them. Right? So gram staining, if it's positive, it's purple. So we're going to see these gram positive bacteria are going to have a purple color, and the gram negative bacteria are going to be pink, because they didn't have that purple stain absorbed to their cell membrane. When they're on a slide together, it's really easy to tell which is which. It takes a little practice. If you're only looking at one kind, you're suddenly going to realize that you have lost your mind and you can't actually decide whether something is purple or pink. Right. So shape of individual bacterial cells, gram stained samples. I will be asking you if you can identify those. That's about all we're going to do with taxonomic affinities for our bacteria, because that's about all we can do in our lab. Right. So those are heterotrophic bacteria. It turns out there's also large taxonomic diversity of what are also called the blue-green algae. And they aren't algae, but as we can see them in ponds, when there's a large number of them, they're actually bacteria. So cyanobacteria, they look a little bit different color than green algae because the pigments that they have for doing photosynthesis are a little different. And they are prokaryotic organisms. And they're mostly going to be found in colonies, and they can actually clump together in these long filaments and big mats, so they can become visible to the naked eye. If they are not in long strands, then they're going to clump together in these little groups. So we're going to see a couple examples of these in lab today. And one thing to notice about them is that the bacterial cells, the heterotrophic bacteria, were very, very teeny cells. They're on the order of a couple of microns. And you may not be able to read it on these slides, but the cells that we can see, these filaments of the cyanobacteria, are closer to 20 microns. So we can see them much more easily. They're much, much larger, right? So we're going to look at a couple examples of cyanobacteria. When we think about bacteria in general, that you want to keep in mind that there are these two different groups, autotrophic and heterotrophic. And both of the groups, when we get enough individuals together, they're actually going to be visible to the naked eye. So a single cell of either of these types of bacteria is going to be too small to see with the naked eye. But when we get a colony of them, whether it's a long filament or whether it's growing together in a glob, we're going to be able to see the colony. Right? So the colonies grow with asexual reproduction. They're going to spread out across whatever surface they're on. So we're going to use these agar plates. Agar is a kind of gelatin, but it's made from seaweed rather than pig's feet. And it's a way that we can grow colonies, large amounts of bacteria in order to study them. We can examine their properties, characteristics. If you take a microbiology course, you're going to be mostly studying bacteria, and you're going to be learning a lot of lab techniques that are going to allow you to distinguish what kind of bacteria it is based on sort of a decision tree of, does it grow on this kind of agar plate? Is it resistant to certain types of bacteria? So there's this set of steps that you're going to be able to do using lab techniques. So left-hand side is just showing that we can do a system of streaking of our colonies to try and isolate ones at a time. We have an activity today where we're going to be able to look at 2 different species of bacteria and see if they are killed by certain antibiotics, in which case you're going to be able to see that this agar plate is clear around this disc. So those little paper discs have been soaked in an antibiotic. If that antibiotic kills the bacteria, you'll see a clear area around it. If that antibiotic doesn't kill the bacteria, you'll see that they grow right up to it. Antibiotic resistance among bacteria is one of our pressing health concerns in general. Before the pandemic hit, it was one of the hottest topics that we have been caring about with human health, because if we don't have an antibiotic, then people with bacterial infections are just going to have to be sick. And it's truly been a medical marvel to be able to treat bacterial diseases with those medicines. On the lower part of this slide, we're showing that when we use different types of agar, we put different substances in it, different nutrients that the bacteria is going to have to use to grow on. We put different substances into those nutrient mixtures, and we have certain types of bacteria or even certain strains of bacteria that are only going to be able to live on certain types of agar. So we can narrow down our choices based on what type of agar they grow on, type of medium. All right, so remember that a single individual bacterium is not going to be visible to the naked eye, but when we have enough of them, they form a colony. Over 1 million bacteria, we're going to be able to see that colony with the naked eye. All right, so if this were a normal semester, I would have sent you home last week with your very own agar plate, and you would have had a chance to put it out somewhere around your house or yard or workplace or horse barn or dog kennel or wherever you think might be an interesting place. And so what ends up growing on those plates is the result of asexual reproduction from spores that are floating around in the air. So fungi and bacteria are both going to be things that are going to grow from those spores. Fungi are going to include yeast. Now, yeasts are single-celled fungi. They're going to grow into these colonies. So these are fungi, right? So molds, yeasts are going to be fungi. And fungi are eukaryotic, whereas our bacterial colonies are going to be prokaryotic. So if I ask you a certain colony, whether it's a prokaryote or eukaryote, you can't tell if it has that kind of cell by looking at the cell. Obviously, if you don't have a microscope, you just have a picture of the plate. So we're looking at the colonies, and you need to be able to recognize what fungal colonies versus bacterial colonies look like, all right? It's not that hard. I learned it pretty quick. I'm going to teach you how to do it. So you should be able to recognize the difference between fungal colonies and bacterial colonies, and there's a couple of clues. Yeasts are going to look like drops of paint or drops of opaque nail polish. They're going to be shiny, and there's a relatively small number of colors that they're going to be, sort of white, pink, yellow, orange, salmon colored, right? So opaque means that light doesn't shine through it. So they're going to be very highly pigmented colors and really shiny and kind of dome shaped on the top, all right? So if we look at these from the side, they tend to be kind of a little blob, like a drop of water or paint. The other most common types of fungi we're going to see on our plates are molds, right? And the molds are going to be fuzzy or furry looking. They're going to stick up off the plate. They're going to often have a very filamentous look to them. All of these organisms are going to grow in a circular shape typically, so that doesn't help you out too much. And the colors are also diagnostic. They're going to be either this blue-green color, or they're going to be black typically, and they may have white, especially around the edges. So there's several different types of molds on this plate as well. So the bacterial colonies are going to tend to be more flat. They're going to be dull and not shiny. They may have a lacy appearance. They're often this sort of bacterial colony yellow, and they're more translucent rather than being opaque, right? So we might have a yellowish colored yeast, but we aren't going to be able to sort of see light through it. We're going to be able to sort of see light through a bacterial colony. They may have a really interesting surface pattern, but it's going to usually look dry, right? So we're going to try and look at a bunch of examples today after I finish lecture, so you have a better idea of telling these apart from one another. All right, so we're going to get into the eukaryotes, and we're going to focus on two groups today, the excavates and SAR. We're going to come back to archaeplastids and uniconts later. In the excavates, we're only going to look at a subgroup. Notice at the top of this table, I have supergroup, group, and subgroup, and then examples. The supergroup is one of those very big picture taxonomic groupings that's wedged in between domain and kingdom, right? So it turns out we actually have a couple levels of categorization now, and they don't have formal names anymore. So for the purposes of this lab, I'm just calling them supergroups, groups, and subgroups, right? To represent that those are a nested set of taxonomic groupings that we don't know what to call them yet. The examples are going to be a common name or a genus name of an organism that we looked at, okay? So we're going to look at the excavates. The subgroup for them is called Euglenozoa, and we have two kinds of organisms we're going to look at, Euglena organisms and trypanosome organisms, and they have somewhat different characteristics, although they share the characteristics that put them into that taxonomic grouping together. Within SAR, we're going to look at the stromatopiles, alveolates, and rhizaria, and we're going to look at two different examples within each one, two different subgroups, all right? Ciliates, dinoflagellates within the alveolates, diatoms and phaeophyta. Phaeophytas are brown algae like giant kelp. And then the rhizaria has radiolaria and foraminifera. So when I'm going through these next few slides, there's going to be a lot of characteristics that we're going to talk about. I have a separate slideshow that has a summary of the characteristics of these groups. So if you're having trouble writing them down as I'm talking, you want to go and look at this Introduction to Eukaryotes tutorial. Right? So there's a bonus slideshow with no irritating narration by me. It has all the characteristics listed out, and it also has a lot more different pictures. All right, our first example, we are in the excavates, and we're looking at our first example, which is trypanosome. So trypanosome organism causes African sleeping sickness. So it's a parasite. It lives inside the bodies of mammals. And in this slide, we can see these little organisms that are scattered in amongst the red blood cells in this sample. So these organisms are very small. They're single-celled. They're a parasite, which means they're sucking up the life out of the host. So they're heterotrophic, right? The characteristics of the excavates include that they have a special kind of flagellum, and that they have sort of a ditch or a groove long ways along their cell, but you can only see that on a scanning electron micrograph. Right, so one of our examples of the excavates is this trypanosome organism. Our other example of the excavates is Euglena organism. Right, we can tell by its nice bright green color that it does photosynthesis. One cool thing about Euglena is that it has this red eye spot, as a photosynthetic organism, it's nice to be able to tell light from dark so that you can swim in the direction that the light is. Now, it does have a flagellum, so even though it's a plant-like organism that does photosynthesis, we can see that it does locomotion, right? So it's an example of an organism that is does photosynthesis and also has the capacity to do locomotion. So when we want to figure out if something is a euglena or not, one thing I clue in on is that it has these parallel sides. You can't really see the flagellum. If it's a fresh live specimen, you can see that it's green and it has the red eye spot. They're relatively small, and their bodies can kind of squoosh, or their cell can squoosh and change into different forms. But when they're swimming, they're sort of long and stretched out like a greyhound. So that's going to help you recognize what's a euglena when you see one. So those are our two examples of the excavates. Now we're going to switch gears to the sarclade. All right, so SAR includes stromatopiles, alveolates, and rhizaria. We're going to start with the alveolates. We have two groups we're going to look at in the alveolates. We have the ciliates and we have the dinoflagellates. Ciliates are characterized by having cilia, which are short hair-like structures that they can use to swim with. And our example organism, it's not an animal, is Paramecium. That's its genus name. So they have this shape where one end is narrower and the other end is wider, and they have kind of a pointy end and a round end. And so those are characteristics of ciliates. They also, if you see them live, they have a characteristic gliding motion. They tend to swim in a spiral as well. And you can see some of the stuff inside them depending on what kind of preparation you're looking at. If they're live, they can be autotrophs or heterotrophs. They have no cell wall, and they're all single-celled. The next group within SAR, the alveolates, we just talked about ciliates. Now we're going to talk about dinoflagellates. Dinoflagellates have a flagellum, and they're going to be able to do locomotion. They actually have two flagella, and they are photosynthetic, so they are autotrophic, and they live in marine habitats, which means that they're a component of the phytoplankton. So phytoplankton means that they're doing photosynthesis, and plankton means that they're part of the group of organisms in the ocean that are some combination of being small and being a weak swimmer, that they are at the mercy of the currents and tides moving them around. So these are quite tiny, they're only single-celled. We have a variety of different shapes. They have this external structure around them. You can see this sort of coating. So that's a cell wall. And their cell wall is made out of cellulose, just like plant cell walls are. So notice this shape that you can see across here. One of their flagella goes into that groove, and it causes them to have a spiraling motion as they're swimming. These little notches at the edge of the groove are going to be a characteristic that you're going to look for. So sometimes when there's a smaller picture, it's harder to see, but there's groove, there's groove. You can see those little notches here and here. You can't see it on all of them, or you can only see it on the ones that are laying the right way. But any picture I might give you, you're going to be able to see that. Right? So they can have fairly rounded shapes, or they can have this kind of pronged shape. The serratium is a very common dinoflagellate. All right, so they are all single-celled. The next group within SAR is the stromatopiles. And in the stromatopiles, we're going to look at two groups, diatoms and phaeophyta or brown algae. Diatoms are also a part of the phytoplankton. So I'm an oceanographer, this is very important to me. Phytoplankton, plankton. All right, phytoplankton. Diatoms have this a variety of different sizes and shapes that they can be. Sort of classic basics diatom example looks sort of like a petri dish or a casino poker chip with those little grooves and ridges going around it. And they're actually more like a petri dish that they have a top and a bottom. And the outside of the cell here, this sort of rigid structure, is indeed a cell wall. And it's made out of silica dioxide. So if you can think about something else that's made out of silica dioxide, it's clear, it's hard, and it is glass. So the light shines through this cell wall, and when we're looking at them under the microscope, sometimes we have to adjust the iris in order to be able to see them well. All right, so this species name is called cyclotella. It's very common. You can see when they're alive. They're a nice glowy green color, telling you that they are doing photosynthesis. They're a really weird organism because they can do locomotion, but they don't have cilia or flagella, and we, the scientific community, do not understand how they do that locomotion. They have no known structure that allows them to do locomotion, yet you can see them gliding along in the water. There's a big variety of structures of diatoms. The easiest way to tell when they're alive is that, bright green, sometimes sort of a golden yellow color. When they're dead and you're only seeing their cell wall, the light is shining through it, and it really looks like glass. So it has kind of an iridescent or rainbowy look to it. They can take on a wide variety of shapes, ranging from circles to footballs to pens to triangles to double footballs. But the thing that's going to be consistent is that when you change the setting of the iris, they're going to look different, and so the light kind of shines through them differently. And overall, there's two shapes that generally they can have. The circle shape is called centric, and the long and skinny shape is called pennate, like a pen that you write with. All right, so we'll see some examples of diatoms. Oh, the other thing to notice is the pattern of holes that is on that test or shell, the cell wall. It looks like a spaghetti drainer or a sieve of some kind. The holes aren't in a specific pattern, but they're all the same size, and they have a recognizable pattern compared to other types of single-cell eukaryotes. The next example group within the Stromenopiles is Phaeophyta. These are brown algae. Brown algae are going to be typically fairly large, multi-celled. They're all multi-celled, and they're going to have this brownish color because they have different types of photosynthetic pigments than land plants do. So land plants are green because they have a photosynthetic pigment that absorbs light and wavelengths that are not green, and it reflects the green. So the name of the pigment molecule in land plants and in the green algae is chlorophyll, and the name of the molecule in brown algae that makes them be a different color. There's a variety of them, but my favorite is called fucoxanthin, and that gives it its particular color. So this is an example of a really adorable brown algae called Postelsia or sea palm. Giant kelp is another example. If you go down to the beach in San Diego on any day, you will find a pile of brown algae all over the place. So they don't have structures like plants in that they don't have specialized cell types. They don't have roots that are going down into the soil. They just absorb their nutrients from the water around them. So they have a structure called a holdfast that is root-like in that it is holding the structure to a rock or a nice hard substrate of some kind. But it's not taking up nutrients, and the cells don't have specialized structures in them. The stipe is like a stem. It holds up the blades. We call them blades and not leaves because they don't have any kind of complex structure. If we look at a cross-section of an algal blade, all the cells are the same kind. There's no differentiation between them. at least to the extent that land plants have. All right, so they do have cell walls. And we see a whole wide variety of examples of body form, but they tend to have fairly large blades and a fairly sort of flat structure, not too frilly or leafy. All right, our last group are the Rhizaria. So within the SAR, remember stramenopiles, alveolates, and Rhizaria. And we have two groups within the Rhizaria, foraminifera and radiolarians. Foraminifera have a cell wall made, again, of calcium carbonate. So their cell wall is made of calcium carbonate. And that means that they're opaque. They aren't translucent. So we're going to go look at microscope slides, and they're fairly large. They're you can see them with your naked eye. So we can't look at a whole foram under the microscope. It just looks like a black blob because the light can't shine through it. So when we look at these on the microscope slide, we're actually going to have a section through them, and they actually look like little tiny seashells when they're sliced through. So they have this globby shape, sort of lobes. They grow in sort of a spiral, and then they have these dots all over them that are in a random pattern. So although the dots look kind of like the diatoms dots, the overall shape of the cell wall is kind of asymmetrical. It's kind of rounded, whereas the diatoms are basically going to be flat. So these extensions that are sticking out from the test or shell or cell wall are pseudopods. So when we think about pseudopods, we often think about amoeba, and we're not looking at amoebas for a couple of weeks, but an amoeba is going to have large, thick pseudopods, whereas our Rhizaria are going to have very thin pseudopods. And they're sticking out of this, they stick out through the little holes in the shell. And our foraminifera are mostly benthic, that is, they live on the bottom. They can actually crawl with their pseudopods, so they can do locomotion. All right, notice that they're not a glowy green color. They are heterotrophs, and they're also going to partially be able to feed by those pseudopods being sticky, and then they can pull the food inside. Here's some more diversity of shapes of foraminifera, but they all have those characteristics of looking kind of globby and spirally, and that the holes that are all over their surface are all the same as one another. They're relatively small, and they're in no particularly organized pattern. All right, our last group in the Rhizaria are radiolarians. So radiolarians have radial symmetry if you look at them from the top. They're rounded shapes, and they also have holes in them, but the holes are much larger. So if we look at the shape and size, they are going to be basically some kind of round shape. They may have spikes sticking out from them. If they're live, they may look green, but that's because they have single-celled algae that live inside of them. And they are going to have these very fine pseudopods sticking out through the holes in their tests as well. And they use them to reduce drag, not to crawl with, because they're planktonic organisms. So they're just basically going to be floating. They have a little drop of oil inside the cell that makes them neutrally buoyant. Right? So they don't use those pseudopods to do locomotion. They basically don't do locomotion. They just drift. Right? But they're heterotrophs, like the forams are. And the kinds of shapes that if they're not just a sphere, they have often shapes that remind me of rocket ships or spaceships of some kind. All right, or a bell shape. Now, of these different kinds of organisms, the ones that have these cell walls, we have a rich fossil record of them. And depending on the type of environment or habitat that different species live in, we can learn a lot about paleoclimate from looking at the fossils that are coming from the seafloors of ancient seas. And we can look at the ratios of different species of radiolarians, of foraminifera, of diatoms, and several other types of these planktonic eukaryotic organisms. And we learn a lot about ancient climate and ancient ecology from looking at those fossilized remains of their shells. All right, that's the end of our lecture today.